Welcome. Aim. What is the dermatophytes? Explain. Name of students Samruddhi Sanjay Patil, Vaibhavi Patil. Roll number 1314. Guidance by Mr. Azad Dhage sir. 2024 to 2025. Dermatophyte. Dermatophytes are a type of fungi that infect the skin, hair, and nails of humans. Definition. Dermatophytes are a group of fungi that require keratin for growth and live in the keratinized surface layer of the skin. General characters. Dermatophytes are cutaneous fungi which infect only the keratinized tissues by liberating keratinase enzyme which helps them to invade into keratinized tissue like stratum corneum layer of skin, hair and nail. It is a group of about 40 related fungi that belongs to three genera. First Microsporum, second Trichophyton, third Epidermophyton. They are restricted to non-viable skin because most are unable to grow at 37 degrees Celsius or in the presence of serum. Many species have particular keratinides, elastase, and other enzymes which make them host-specific. Several are capable of sexual reproduction, produce ascospore, thus belongs to genus Orthoderma. In skin, they produce hyaline, septate, branching hyphae or chains of arthroconidia. Efloxum is the only pathogen in this genus which produce microconidia. They are highly contagious and frequently transmitted by exposure to shed skin, scale, nails, hair containing hyphae and conidia. They remain viable for long periods of fomites. Classification according to shape and site of infection. First, Microsporum. Spindle shaped, infect skin and hair. M. canis, M. gypsum, M. gallini, M. nanum. Second, Trichophyton. Pencil shaped, infect skin, nail and hair. T. rubrum, T. tonsurans, T. mentagrophytes. Third, Epidermophyton. Club shaped, infect skin and nail. Efloxum. According to habitat. First, anthropophilic habitat in human body. Some Trichophyton species. Efloxum. Second, geophilic, usually habitat in soil. M. gypsum. Third, zoophilic, usually habitat in animal. M. canis, dogs and cats. M. gallini, fowl. M. nanum, pigs. T. equinium, horses. T. verrucosum, cattle. Immunity. Trichophytid reaction. Trichophytin is a crude antigen preparation that can be used to detect immediate or delayed type hypersensitivity. Chronic non-inflammatory dermatophyte infection. Poor cell-mediated immune response to dermatophyte antigen. Immediate type hypersensitivity with elevated IgE. Allergic reaction. Dermatophyte, usually vesicle and often in hand. Trichophytid reaction, markedly positive. Clinical findings. Dermatophytosis is classified according to site of involvement. First, Tinea capitis, infection in scalp and hair. Tinea barbi, infection in beard area. Third, Tinea corporicis, infection in the trunk. Fourth, Tinea crucis, infection in groin, inguinal region. Fifth, Tinea manum, infection in hand. Sixth, Tinea unguium, infection in nail. 7. Tinea pedis, infection in foot. Tinea capitis, dermatophytosis or ringworm of the scalp and hair is called tinea capitis, caused by trichophyton or microsporum. Pathogenesis, hypal invasion of the skin of scalp. Subsequent spread down the keratinized wall of hair follicle. Infections begins just above hair follicle, grow downwards on non-involving area as the hair grows upwards. Production of dull gray, circular patches of alopecia, scaling and itching. As the hair grows out of the follicle, first microsporum, hyphae production chain of spores forming sheaths around the hair shaft, ectothrix. Second, trichophyton, hyphae produce spores within the hair shaft, endothrix. Hair become weakened, typically break easily at the follicle opening. Third, zoophilic species induce combined inflammatory and hypersensitivity reaction. Carry on. Fourth, Trichophyton sciolini acute inflammatory reaction of hair follicle leading to formation of scutula. Favors. Pathogenesis. Fungal metabolites, enzymes, antigens diffuse through the viable layers of the epidermis. Erythemia, vesicle information, prutesis, lesion, expand centrifugally and active hypal growth at periphery. Tinea unguium, onychomycosis, may affect toenails or fingernails, but toenails infection is particularly common which follows tinea pedis caused by trichophyton and epidermophyton. Risk factors: diminished blood supply. Second, humid moist environment. Third, perspiring heavily. 4, barefoot in damp places. 5, DM immunosuppression. Course of disease: itching between toes, development of small vesicles, rupture of vesicle and discharge of a thin fluid, skin of the two webs become macerated and peels, cracks appear and secondary bacterial infection develops. Vein become conic, peeling and cracking are accompanied by pain and pruritus. Laboratory diagnosis. Sample. First, skin scraping. Second, nail scraping. Third, hair plucking. Collection of samples. First, skin from the margin of the lesion with the scalpel. Second, nail. Deeper part is collected and superficial part is discarded. Third, hair plucked by fine forceps. Microscopic examination.